Welcome back to Brain Bites. Today we're taking a closer look at a subject that affects our health, lifestyle, and everyday decisions more than most people realize. We're cutting through the noise today, separating the absolutely mind-blowing reality of CRISPR gene editing from, the wild science fiction hype that usually surrounds it. It's a story about having the raw power to literally rewrite the code of life itself. But it's also about the massive, very real hurdles, from staggering price tags to intense geopolitical rivalries that stand between a laboratory breakthrough and a treatment at your local doctor's office.
Okay, let's dive into this. Here's our map for today's explainer. First, the weight of a Nobel. Second, how CRISPR actually works. Third, real cures and high costs. Fourth, AI hype versus biological reality. Fifth, funding and global competition. And finally, number 6, the ethics of designer babies.
So, Part 1. The Weight of a Nobel and Jennifer Doudna's incredible journey. To really grasp CRISPR, we've got to start with the mind behind it. Jennifer Doudna grew up in Hilo, Hawaii. And get this, she had absolutely no scientist in her family. Fast forward a bit, and she's receiving the Nobel Prize. Literally sitting right in her backyard garden because, it was the middle of the global pandemic. And with that ultimate prize came some immense pressure. Her husband actually told her, "Your main job now is being an ambassador for science." And she totally embraced that. She went on to found the Innovative Genomics Institute. Now, this isn't just your standard academic research lab. It is a massive economic powerhouse squarely aimed at curing diseases. It's already spun out 31 companies, created 2,500 jobs, and reached a staggering $9 billion valuation. Just incredible.
Moving right along to Section 2, how CRISPR actually works. Let's break down the science of editing.
The craziest part of this whole explainer is that CRISPR didn't even start as a way to edit human DNA. It actually began as a study into the remarkably adaptive immune system of simple bacteria. Basically, it works like a giant puzzle. Step 1. When a virus attacks a bacterium, the defending cell grabs and saves a tiny fragment of that viral DNA. Step 2. It turns that fragment into guide RNA, which functions almost exactly like a tiny genetic GPS. Then step 3, if that specific virus ever tries to attack again, a protein called Cas9 steps in, acting like molecular scissors. It pairs up with that GPS, hunts down the invading virus, and literally snips it apart. The absolute game changer here was when Doudna and her team realized they could reprogram this bacterial GPS and scissor system. They figured out how to dictate the exact coordinates, directing those scissors to target and edit specific sequences inside human DNA. Just like that, editing the human genome wasn't just science fiction theory anymore, it was reality.
Okay, Section 3. Real cures, high costs. Taking this from the lab straight to the patients.
Professor Doudna feels this intense, daily urgency because there's still a massive divide between making discoveries in a lab and actually getting those therapies into suffering patients. We're talking about real lives here. Take Victoria Gray, for example. She was the very first patient with sickle cell disease treated with CRISPR in the US, and she went from a lifetime of debilitating pain to essentially being completely cured. Her words on this are just so powerful. She said, "I want to reach others who feel like no help is coming. We're coming." Wow. It really showcases the absolute life-changing miracle of these therapies.
But we do have to sober the narrative up a bit here. $800,000. That was the eye-watering price tag for a personalized CRISPR therapy just recently in 2025 for an infant known as baby KJ who is battling a lethal metabolic disorder. Now, the great news is he's currently crawling, walking, and thriving, which is an absolute triumph. But let's be real, $800,000? That is simply not a scalable public health solution. The reason it's so incredibly expensive right now is that scientists have to use what's called an ex vivo process. That means they take cells out of the patient, carefully edit them in a lab, and then transplant them back in. So, over the next few years, the entire industry absolutely must transition to much more affordable in vivo treatments, where the CRISPR molecules are delivered directly into the patient's body to do their job right there on the spot.
So, with costs being that high and the biology being that complicated, the natural question everyone is asking is, can artificial intelligence speed up these miracle cures? Before we dive into that answer, if you're getting value from this video, a quick like would be greatly appreciated. It helps support the channel and enables us to bring more high-quality content to viewers around the world. All right, now let's see what the Nobel Laureate actually thinks about AI's role in biology.
That brings us to Section 4, AI hype versus biological reality.
We are witnessing this massive tension right now between Silicon Valley promises and hard biological reality. On one hand, you have tech executives at OpenAI casually suggesting they should get drug royalties if ChatGPT invents a cure, and Larry Ellison boldly claiming AI will just cure cancer in a 48-hour window. But Doudna, she's much more grounded. Her reality as a biologist is that biology is incredibly, mind-numbingly complex. Sure, AI is fantastic at summarizing massive data sets and writing up reports, but actual ground-up innovation, that is still very much in the human domain. To put this complexity into perspective for you, it's been 26 years since the human genome was sequenced, right? Yet we still don't even understand the function of 40% of the genes in a simple single-cell bacterium. So yeah, we aren't just going to simulate our way out of that kind of deep complexity overnight.
Let's jump to Section 5, Funding and Global Competition. The global science race is definitely on.
As if those biological hurdles weren't enough to deal with, we also have to look at the economic and political realities threatening this whole field. Now, we're strictly reporting the numbers here, but data shows that under the Trump administration, federal funding cuts created some pretty significant uncertainty in the scientific community. Over a 3-year period, new grants from the National Science Foundation actually dropped by 24%. On top of that, roughly 25,000 scientists and staff, which is about 20% of the workforce, ended up leaving federal research agencies. Those are some stark figures, and they carry real long-term implications for our research capabilities.
So why does Doudna argue that this matters so much? Well, first off, the economic return on investment for science is pretty undeniable. Historically, every single dollar invested by the NIH yields about $2.50 in economic benefit. It's huge. Second, if the US pulls back on this funding, well, the global economy abhors a vacuum. There's a very real risk of ceding our biotech leadership to China, which has been rapidly accelerating its state-backed investments in science. And finally, when it comes to domestic political movements, like MAGA, for instance, Doudna maintains a very firm, objective stance. She argues that broad health decisions absolutely must be rooted in clinical data, empirical testing, and rigorous science, rather than just rhetoric.
All right, finally, Section 6, The Ethics of Designer Babies, Altering Human Destiny Itself.
You know, this isn't just some far-off theoretical debate anymore. Following the massive international outcry back in 2018, when a Chinese scientist claimed to have created the world's first genetically edited babies, we really have to confront our sudden power to alter human evolution. Some folks in Silicon Valley might love to push the idea of breeding super babies by selecting for complex traits like intelligence or height, but the actual biology says something completely different. Those complex traits aren't just controlled by one single easy switch. They are controlled by thousands of interacting genes. Tweaking them for so-called enhancement risks some massive unintended biological consequences. Because of this, Doudna strongly advocates for a pragmatic two-buckets approach. In bucket number 1, we use CRISPR specifically to target devastating diseases that have a clear, well-defined genetic cause, things like sickle cell. But in the second bucket, that's genetic enhancements, which carry extreme risk and, at least in her view, really shouldn't be touched anytime soon.
So what does this all mean? It really boils down to one profound question. With all this immense power now in our hands, is our biotech future headed toward a literal paradise of cured diseases, or more of a dystopian Gattaca scenario where humanity gets completely divided by genetic enhancements? Well, Jennifer Doudna remains an optimist. Despite the incredibly heavy burden of her discovery, she genuinely hopes we land closer to nirvana, using this miraculous tool to benefit as many people as possible. Thank you for spending your time with us today. If you enjoyed this video, please like, share, and subscribe to Brain Bites. We'll be back soon with more fascinating topics, expert insights, and stories that matter.